spaced, I don't know, every 8 feet or so with multiple leaders to fill that in. So there's many advantages to these fruiting walls. I could talk to you a lot more about tree fruit agriculture over lunch, but they do lead to reduction of labor, spray and land, and increased quality yield and enable mechanization. But they're not very feasible for every type of fruit tree. Really only the ones that are quite profitable or you could sell for a lot, like apples, because it takes a lot of money to install and set up trellis and do extensive pruning and labor. So it's not easy for everything, which is why I'm trying to use my interest in developmental biology to figure out a comprehensive understanding of molecular and genetic control of tree branch orientation, essentially for breeding strategies and management practices. So for this reason, I got interested in lazy very long ago when there were just a few papers about it. And it came from my postdoctoral research project where I helped identify that this this pillar peach tree here actually has this architecture because it has a homozygous mutation in TAC1, which we then found two papers, maybe 2 or 3 on TAC1, where it has a narrower tiller angle, and connected it bioinformatically to lazy, which we've heard all about. TAC1 has the opposite phenotype. You have much narrower lateral branch angles. So I began studying lazy Arabidopsis back in 2012, 2013, and in between I did a lot of tree stuff. So it took a while to get a paper coming out that connected the 2 genes, but I am sort of working on the integration of these 2 with light and gravity to help the tree fruit industry. So part 2, is it useful to be lazy in the orchard? So lazy, the Arabidopsis version, you see has a nice wide angle phenotype, and that's something that could really assist with planar fruiting wall systems. So we silenced lazy in a tree. And I did first come about studying lazy from a peach tree, but peach is not transformable. It is a bit harder to work in trees than Arabidopsis. So we silenced it in another prunus tree that is also useful for agriculture, plum, Prunus domestica. And this is all work that I've been doing for about 12 years, and it is now just available on bioRxiv because trees take a while to grow and we want to characterize them. We did some training studies, I'll talk to you, and wanted to look at fruit. When lazy was silenced in plum, there were wider petiole angles. You could see the leaves drooping here. And in the lines that were silenced, so all but number 2, there were wider branch angles and outward orientations. So this was very exciting. So when I was at the USDA in West Virginia, in Chris Dardick's lab, I put my first field tree planting in. And within the first year, right before I moved to Michigan State, we saw some very promising architectures. So lazy trees grown out in the field had that beautiful right angle phenotype. Maybe we could engineer plums or other fruit tree species to not even need trellis, to just have, or be very easy to train in trellis with this right angle phenotype. So then I moved to Michigan State University, started my lab, and one of the first things I did was I worked with a horticulturist there that specializes in training sweet cherry, and we put in a planting where we trained lazy trees in this espalier method to see is it going to be much easier. I was very excited about it because of this beautiful phenotype, and we've learned a lot of things in the past few years. So fruit trees are vegetatively propagated. Many of you know that maybe through grafting. So for this study, we took buds from lazy transgenic trees, we grafted them on rootstock and grew them out. We also looked at T1 regular, right out of the tissue culture lazy plants and saw how they grew on their own roots. And while this study was going on, we also observed them in pots in the greenhouse. There were quite a few things we learned from studying lazy in plum that I think may be useful to understanding the gene in other contexts. One of them that was surprising right away, so if anyone has ever done grafting, usually this is not I didn't have a good example of a graft, but if you, cut and paste your bud onto a rootstock, and it would normally grow out and grow straight. So that's why you can graft and have new trees, because the new bud, it's as if it had lost apical dominance, will grow up. But when we grafted lazy onto our rootstock, it didn't want to grow up. It completely lost its ability to grow upward when that bud was forming. So it just grew out and it grew down. That suggests that lazy 1 is important for establishing this directional shoot growth early on, before there maybe is gravity sensing, or when just gravity is being sensed in the developing bud. And that was very surprising. Also, looking at our root-bound semi-bonsai trees in the greenhouse, you could see there's a lot of wild growth. It's not just perfect up and out like we were hoping. It's a bit wild and wiggly. And I always think of this as maybe this is a battle between light and gravity. So there are other lazy genes in plum, and they could be, partially involved in still maintaining some upward orientation, but it's sort of like wiggling for light, and in the process, I think getting lignified. Out in the field, these are very beautiful ornamental trees. You can see they might look similar to some other ornamentals, weeping. Okay, so the espalier study. So we put these very tiny budded up trees in the field, let them grow for a year, and then we started training. So after 1 year of growth, right when it's entering the second season, this is what it looked like before we started doing any pruning and training. And we have kind of this wild growth here. I want to point out that just like you saw with the lazy grafts, there's a loss of apical dominance. We could not get, even though this, was grafted way down here and grew up, we could not get the lazy shoots, even the primary leader, to ever grow up. It was a lot of tying. And there's a lot of wiggle going on here, where I think that's, I don't know if it's a circummutation phenotype getting frozen by the lignification or what's going on. And you could continue to see this as we trained them out. Needed to do a lot of tying to get that to go straight up. And just continuing the study is very obvious. This is our standard control. We have shoots that are breaking because of the loss of apical dominance when we lay it down, and they go straight up. But lazy, of course, goes either horizontal or it curves. And this continued, and it's beautiful architecture. I think the jury is still out on whether this is beneficial for fruit tree ag because of some of the slides I'm about to show you. There were some unexpected but very interesting findings about lazy and plum. Whether it relates to lazy function in other systems and herbaceous species, it's possible, but it's just not visible in those. One was we saw extreme chlorosis in several of our lazy lines, and this happened kind of mid-season, as if it took a high light accumulation. Very chlorotic, there was reduced chlorophyll, and there was a reduction in leaf photosynthesis. We also noticed some reproductive phenotypes. First was the flowers. Here, standard flower, one carpel right here, one ovary at the bottom. On occasion, in stone fruit, like cherry and plum and peach, you will have two carpels or two pistils, and that's usually connected to environmental heat stress during the developmental process. But here, for 2 years in a row, we saw a tremendous increase in the number of lazy plum trees that had additional carpels. And most of the fruit from flowers like this will abort. Occasionally, we will have a couple that are kind of two-lobed. Okay, so some of the lazy fruit were normal, but some were not. And this was very interesting. It took until year 10 to learn this, so that was 2 years ago. Towards the end of ripening, they would explode, and they would explode at this suture. And it did not have to do with having all of a sudden an excess of water, which could cause cracking in cherry. What when they would explode, it also broke the pit. Here, this shows that there was an uneven ripening. So a couple slides ago, this is fairly even. Maybe this is a little darker. But sometimes, look, this is quite a gradient going on. Green blowing up kind of in the middle. And some of the the fruit from the lazy trees that split also had split pits. And those pits had seeds that were germinating. So what I mean by split pit, so normally this is your fruit, you open it up and you see a pit. That pit is a hard lignified tissue on top of your seed, which looks like an almond. So this is what normally would happen. If you opened up these fruits, you see the seed immediately. The other side of the pit would be on the inside of this fruit. You can see there's a little bit of germination happening here. More split pit, it's starting to grow out here. So some lessons from 12-year-old transgenic plums about lazy. Lazy silencing caused outward wandering, wiggling branches. That may relate to the lignification in a woody tree that's capturing circumnutation phenotype, auxin movement phenotype. I'm not really sure. And whether lazy silencing is beneficial for tree fruit management, we could talk about that later. Probably it's not good for growers to have chlorotic trees with exploding fruit. But that didn't happen in all of the lines. So lazy silencing induced chlorosis, reduced leaf photosynthesis. So do these connections relate to lazy and other species? Maybe, but we could discuss that further. And it may have a role in things beyond gravitropism because we saw those reproductive phenotypes. So this is the lazy story. I do have another 15 minutes, so I hope your stomachs aren't grumbling too much, but I'm going to keep talking about fruit. Oh, with this, these different phenotypes in lazy, you could correct me if I'm wrong, but I don't think it's fully known what the ear domain and the C-terminus of lazy does. Maybe this has something to do with it, some reproductive role. Okay, I'm going to switch into a related gene and I'll explain why they're related, one called weeping. So weeping isn't always a bad thing because in studying these weeping peach trees, which I was doing for the same reason I was studying those very narrow TAC1 pillar peaches for tree fruit agriculture manipulation, we identified a new player in auxin movement and connected to gravity response. So this is a weeping peach tree. This is our newest paper out of the lab. I don't remember how many years of work, 8 or 10. So when you work with fruit trees, it's very, very rewarding when you get a publication. There's a lot of work in them and there's a lot of people involved. So these trees, they grow downward no matter what. And this gene, the weep gene that's deleted in here, is very conserved throughout plant species. This is the connection to lazy. There's a common epistatic relationship. So published a few years ago that TAC and lazy, double mutants look like lazy. So lazy is epistatic to TAC. And years before that, the breeder I used to work with, the peach breeder, identified that weep and TAC mutants, we didn't know what genes those were, look like TAC. So TAC was epistatic to weep. So there is some connection. Okay. So it's really nice to be able to study things in Arabidopsis. And many of you are studying lazy in Arabidopsis. I am too, as well as TAC. But unfortunately, it was not a good model to study weep. Even though it is expressed, and you could see the Gust reporter, there is no shoot phenotype in weep mutants. And I looked at many. The lack of a phenotype might have something to do with it not being woody. It also might have to do with 7 different mutations in the SAM domain, despite this protein being very highly conserved across plants. Brassicaceae have mutations in the SAM, and some of these are where there are phosphorylation sites. So we've been studying it in peach for many years. So they always want to grow downwards, and this is the same is true for if you gravity stimulate them. So they do not have a normal shoot gravitropism response. You lay the plants on their side. Normally the shoot tips after a few weeks will grow up on a standard. You'll have new growth growing up. If you lay it on its side, all new growth goes down. If you grow it upside down, so these were pulled out, cut back, turned upside down. All new growth on standard, the standard tree, does grow up, but all new growth on weep always grows down. And if you flip it over, and this is actually was more insightful than we realized at first, it's frozen. So this weeping phenotype, it's stiff. So when we're able to flip the trees back over, they held in that position. Interestingly, Arabidopsis, so it doesn't have a shoot phenotype, but it has a root phenotype. They have narrower roots. Oh, this is pretty small here. This is all published, though. Narrower root angles, and we found that the weeping peach trees also have narrower root angles. So there is some role in roots. And there is a gravitropic role in roots. The lateral roots of peach trees have faster gravitropic responses. The primary do not, and that may be true because of its function is more lateral root related, or it may be that we did not have a larger sample number. This was actually quite a complex experiment that we did about 3 times with very big root boxes, and trees are a bit challenging. But seeing that root gravitropism phenotype in weep roots was pretty exciting. Okay, so there, weep does tie into gravitropism, shoot, and root. So back to the peach shoots. Why are they not exhibiting these normal shoot gravitropism responses? One question is, well, do they know which way is down? Maybe they don't. Can they sense gravity? So we sectioned shoot tips, and we could see we positioned them and fixed them in plastic, sectioned them right away. You can't do live imaging in peach to see amyloplasts, but we do know their original orientation and found that in the weeping peach, the amyloplasts did sediment normally, and they were there. We also looked at basic anatomy because you can't have a certain structure without changing internal structures, without changing cells. There was no big difference in anatomy. There were no differences in lignin or cellulose localization. We were thinking maybe they're reversed. There was nothing obvious, or with a biochemical assay. And there also weren't strength differences. Did that pop up? So they're not weeping because they're weaker. And you could actually see they're not weeping because they're weaker because of, it was a few slides back when we flipped the tree back up. It was pretty stiff. There were slightly smaller cells in the shoot tips, and this took a lot of sectioning and a lot of different ways of sectioning to figure out. Ultimately, we did plastic sectioning for this and looked at area and cell length and cell width in regions from a certain, you have to get the shoot tip at a certain spot and a certain type of branch, and found that there were slightly smaller cells in the weeping trees, and there was no difference between top and bottom. So adaxial, abaxial side differences were found in standard. So as you know, upward growth, think bending tropic responses, you know you're going to have longer cells on the bottom, shorter on the top. And that is what we saw in the standard, but we did not see any differences in the weeping. Okay, so they can sense gravity. There wasn't huge anatomical differences, whereas weeping acting. So, you know, downstream in Courtney went, we have auxin movement. So we tested if these can respond to asymmetric auxin applications. This is in the greenhouse. Standard tree, weeping tree. You could see their shoot tips. Put a little lanolin with some auxin right below on each. As expected, the standard reoriented upwards. This may have had to do with also development of tension wood and not just or not necessarily elongation because they are woody. And the same thing occurred in the weeping tree. So auxin on the bottom did cause it to bend up. We also did this in shoots that you could cut to see it a little bit better, put it in water and did auxin applications, and they bent down. And just FYI, sometimes I use a red leaf weeping tree. It has red leaves and red stems because it's a good contrast. We just have that segregating. It has nothing to do with the weeping phenotype. It's just that we also have red leaf, red stemmed weeping trees. Okay, so these weeping trees that have a mutation in weep can respond to asymmetric auxin application just like standard. So that suggests that weep must function downstream of amyloplast sedimentation, but upstream of asymmetric auxin localization. So next, we did an RNA-seq study taking advantage of how nice and thick our peach stems are, or peach shoots. So we cut them in half, weeping and standard, did the top 2 internodes where you already have that arch right there, and looked at gene expression differences between the top and bottom. And what we found was there were flipped expression patterns of several genes between the upper and lower tissues of each genotype. So meaning in standard, gene X had a lot more expression in the top, but in weep, it had a greater expression in the bottom. So in this heat map, red, so this is standard on the left, weep on the right. You could just kind of see the big contrast. Red was more expression in lower tissues, blue more expression in upper tissues. And what was really noticeable in some of the genes that were differentially expressed between top and bottom and then compared between genotypes was that many of them had to do with auxin. There are lots of auxin-inducible sours, PP2Cs, and AuxIAAs that were notably reverse expression. So down in the blue is more expression in the upper tissues in a weeping tree, while more expression in the lower tissues in the standard. And we know the standard also has slightly longer cells and is growing up. So collectively, this showed that auxin localization is flipped in the weep mutant, and weep is very likely regulating auxin transport because of all of these genes that are auxin regulated are regulated in the wrong place. So the conclusion from this is that the weeping peach trees are growing downwards because they have flipped auxin localization patterns in their shoot tips. And this just sort of diagrams that out. Normally your pins are on your bottom and your auxin moves this way and shoots would bend up. Or in the case of branches, they would just grow upwards at an angle. But in the case of weep, you have the sours and the pins up on the top and a higher auxin on the top. And so weep regulates auxin movement in response to gravity because this is, they can sense gravity and this is connected to the downward growth. This may be true in other species. Weep is expressed in shoots in Arabidopsis, but maybe isn't easily detectable or there's redundancy or different interacting partners because of the mutation. And this role may also rely on multimerization or oligomerization of weep through the SAM domain, which Lucia Strader and her graduate student, Jeffrey, helped us figure out. So the wheat protein actually forms multimers of 7 or 8 proteins. So something very cool. This was from like one of those serendipitous accidental experiments. So weeping trees, if you accidentally let them start growing in your dark cold room, when the cold room fails and it warms up, you'll get etiolated growth. So we keep trees in the cold. When we're growing them in the greenhouse, we rotate them through cold for a fake winter because the vegetative buds need to go through cold in order to start growing. But they only need a certain amount of cold. So if they're in the cold room for a very long time and you don't have room in your greenhouse and you keep them in there for a long time, they will eventually start to grow, especially if it accidentally warms up. And we found that both in the red leaf and the green leaf weeping trees, when they started to grow in that dark cold room, they grew straight up. This was also true with lazy. And this right here suggests that weep is only needed for this auxin gravitropic response movement in the light. So it only influences shoot architecture in the light. So putting these two things together, a couple conclusions. Lazy silencing phenotypes in plum may be valuable for understanding function. If anyone else has questions that you think looking at a tree system could help answer with this gene, I would love to work with you. Lazy may have a role in auxin transport outside of or something else outside of tropic responses. So we could see from the fruit phenotypes. Weep is, I believe, a new player on the auxin transport team that extends beyond trees. And shoot function of weep in plants may not be apparent because they're herbaceous. We did see the root function in Arabidopsis and also the wheat homologue in barley EGT2. Those mutants have very narrow root phenotypes. So things I'm going to be working on currently in the future. I'm looking at common and distinct upstream regulators of TAC lazy wheat. So transcription factors and what triggers them. And I'm very glad I'm doing that because I think half of this room is working on the downstream of lazy. So you guys can provide all of the information about what lazy is doing mechanistically. And I'm doing this transcriptional regulation in trees and Arabidopsis and looking at protein interactors for TAC1 and wheat. And I'll be applying this knowledge to tree fruit agriculture over the next 20 or so years. So thank you. Quick acknowledgement slide. The